This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 7, recorded May 13th, 2011. Hi, everybody. I'm Vincent Racaniello, and this is TWiM, the podcast that explores unseen life on Earth. Joining me today from San Diego, California, Elio Schaechter.
Hi there.
Welcome back.
How are you?
So you here for your second TWiM. You must have liked the first one.
I sure did.
Good.
I had a great time.
Excellent. Good to have you back. Also joining us today, we span the coast today from Princeton, New Jersey, Cliff Mintz.
Hello, Vincent.
How are you, Cliff?
I'm good.
Is everything treating you well?
Absolutely. Can't complain, but I will.
I'm sure. I've known you for many years, and one thing you haven't not done is complain.
Exactly.
But that's fine. Also joining us from the Medical University of South Carolina, Michael Schmidt.
Hello, Vincent. How are you this afternoon?
I'm very well. This is my third podcast of the day. We did TWIP and TWIV and now TWiM, the vagaries of scheduling.
You're saving the best for last.
The best for last. This is amazing stuff here. I think it's great, and I can learn new things at my age as well. Now, today is not only Friday the 13th, which some people feel suspicious about, but it's podcast number 7 of TWiM. And what they say in the podcast business is that most podcasts never get past number 7.
Hey.
So here we go, because we know we're doing number 8, because we're actually doing that in New Orleans next weekend.
You may be jinxing New Orleans, Vincent.
You know, someone told me that there's some water problem down there. Is that right?
That's what we've heard.
I think they're thinking about releasing, the levees upstream so New Orleans doesn't go under.
Wow. Well, that could be a problem. There are a lot of people going to New Orleans for this meeting. But, well, if we don't do TWiM in New Orleans, we will certainly do it from here again the next week. But we will get past number 7. That's my point. We will go on for a long time because there's an awful lot to learn. Today, we also were expecting Margaret McFall-Ngai today, and maybe that's her ringing. Let's see. Hello, Margaret.
Hello.
Welcome back to TWiM.
Thank you.
Yeah, so we would like to know if winter is over in Madison.
Well, the trees are not in full leaf, and it's raining. But otherwise, it's pretty close.
Snow's gone?
Snow's gone, and we've got a flower here and there.
Very good.
It's unbelievable.
All right.
How I miss those days.
Do you? Okay, we have our full crew here. We have Cliff, Michael, Elio, and Margaret. And we have 2 cool stories to talk about today. So let's get ahead of that. Let's get going. The first is a paper in Molecular Cell that Elio brought to our attention. And it's all about the E. coli extracellular death factor, EDF, and its effect on toxins. So this is new to me, Elio, this whole toxin-antitoxin system in E. coli.
Well, it's not just in E. coli. It's in about half of all the bacteria that have been looked at.
Half of them.
Yep.
And what does it do?
Well, let's back off a minute.
Sure.
This phenomenon of toxin and antitoxin-induced death started with plasmid work. People found that when bacteria lose their plasmids, they die. And this was studied in great detail by a bunch of people, including Michael Yarmolinsky, who, among others, found out that this is due to a very interesting module, genetic module, 2 genes, one of which codes for a toxin, and the other one codes for an antitoxin. So when both are made, the cells are happy. And these are encoded by a plasmid. This is true for low-copy plasmids like FP1, R100, and so forth. Now, the other protein is an antitoxin. Now, the story is the following. The toxin is stable, and the antitoxin is less stable. So if you stop making them, Imagine the cell stops making these two proteins. The antitoxin is going to disappear. The toxin stays around and kills the cell.
And that explains this observation that if you remove the plasmid, that the bacteria die, right?
Right.
Okay.
Right. And Yarmolinsky gave this module a very fine name. He calls it the addiction module because the cells better become addicted at having the plasmid around.
Mm.
So that's a wonderful story that's been around for a while. What is more surprising is that people have found chromosomal modules. In other words, nothing to do with plasmid. The story with plasmids makes full sense. Very logical, very simple. But why would you find a toxin-antitoxin system in bacteria? Why would bacteria want to kill themselves?
Mm-hmm.
And this has become a big subject. This is really widespread. You find papers practically on every issue of the journal because they're found, as I said, in so many different bacteria. In addition, each bacterium doesn't just have one module, it has a whole raft of them. E. coli has, I don't know how many, 5, 6, 7. Mycobacterium tuberculosis, if you do genomic studies on it, has something like 80 of them. And many bacteria have a whole collection of different modules that work in a different way.
And these are not just in lab strains, they're in clinical isolates as well?
They're in clinical isolates, right. And they are even found in obligate endosymbionts. So what's all this about? I mean, this is really a new story. And I'll tell you what I know about it in general, that when it comes to the molecular details, there's a lot of beautiful work done, really elegant work. When it comes to the interpretation of the data, to the understanding of why bacteria have evolved this, we are really in the dark ages. There are 9 different models or theories that have been proposed, and they all make sense, and maybe they're all 9 are correct, but the simplest one, I just mentioned the simplest one, then we can go into mechanism a little bit later, is that this is a altruistic form of suicide. These systems are not expressed normally during growth. They're expressed when bacteria encounter bad conditions like starvation, oxidative stress, antibiotics, high temperature. At that point, bacteria stop growing and tend to sort of be starving in a way. And so the idea is if some bacteria die, they'll provide food for the others. So like committing suicide to feed your neighbor. Well, that's one theory. There are many others, but we'll go into that.
So in that sense, it's sort of like apoptosis in
exactly. Yeah,
mammalian cells.
It's called programmed cell death. The word apoptosis is not used because it's so specific in eukaryotic cells. But programmed cell death is what this is called. Yeah.
But I think Elio brings up a really good point by raising the issue about tuberculosis. 80 genes in the economic budget of DNA replication in an organism that grows extremely slowly, there must be some selective advantage for keeping them there.
Right.
And the second thing I think we need to think about is while we're arguing that they're functioning when things get bad or things get different, and that's the argument I think I'd like to make is these things are there so the bacteria can respond to the ever-changing environment. It may not necessarily be stress-related or nutritionally related. I think it just simply has to do with life. As life changes, in the case of tuberculosis, consider it lives out in the environment, and then it becomes this facultative intracellular parasite growing very slowly in a very protected niche where the host begins to wall it off and make a granuloma. And just imagine all the environmental perturbations this microbe must be perceiving as it's living in a chronic granuloma. And I think what these toxin-antitoxin paradigms are teaching us about bacterial gene expression is it's not as simple as the lac operon was made out to be. And the lac operon is really not all that simple. It's a pretty elegant control system. And I think many of our colleagues who are studying apoptosis and actually using apoptosis to as a potential molecular therapy for cancer, really need to take a pause and look at some of these studies because, going after apoptosis.
Let me interrupt you and tell you that the people who work in this field routinely asked to participate in symposia on apoptosis. So this has had some good penetration, let's say, and it's related to that, but nobody knows how. So, you're right. This is all a way of looking at biology, which is relatively new.
Do we know that these toxins, antitoxins, are actually made in a TB-infected host, as Michael was suggesting?
That's a great question, and it points out what's missing in all these studies. These studies are really, as I say, very elegant when it comes to laboratory work. However, if you want to relate them to what happens out in the real world, there is almost nothing done on it. I coined a new phrase, in naturam veritas. In nature is the truth. How do you like that?
I think it's spot on.
Anyhow, I think that on the other hand, it's so much harder to do studies in a natural environment than it is in a test tube, so that I don't blame him for not being ahead of it. But really, right now, there is the studies in nature are lagging sadly in this field, and so Michael, you can be absolutely right in what you're saying about TB, and nobody's going to contradict you.
That's generally the safest way to approach it, and I think the really neat thing about these addiction molecules, I like that term, the addiction molecules, because the addiction molecules are adapting the population, if you will, to a particular set of circumstances. And then as those circumstances change, depending upon which addiction molecules are expressed, and I would predict that in future we're going to see almost like we saw with the sporulation system, where there was a temporal cascade of how these things are made. There may be some triggers akin to the catabolite repressor type control mechanisms, where they're sensing energy charge of the cell through cyclic AMP, and all of these other really elegant genetic systems that are eminently testable in vitro, but extremely challenging to test in vivo in a complex host dynamic. I think what we're going to have to do is tease it apart in vitro, and then once the mass spec technology gets down to the bench level, we'll be able to move it and ask Elio's really important questions about what is happening in nature.
All right. Let me add a little bit to the mechanism, though. Can I do that?
Yes, of course.
Because it'll give you an idea. So far we've only talked about this altruistic suicide. There is more to it. If you insert these modules into a series of genes, and happens to be in an integron, but it doesn't matter. In a series of genes that can be excised, the presence of the module will ensure that they don't get lost, because when they get lost, you lose the module, the cell will die. In other words, this is a way that a set of genes may have learned to make sure it's retained and not excised from a chromosome and lost. And this has been used industrially. It's now used for plasmid maintenance. When you engineer a plasmid and you put it into a bug and you want to grow a 100,000-gallon fermenter with it, it behooves you to make sure that the plasmid isn't lost. Well, if you take a plasmid and you engineer into it a particularly good addiction module, the plasmid won't get lost. So there's more to it, and it's really fascinating.
Elio, could this be a way that we still have bacterial species? Because one of the things that's always puzzled me about evolution is, why is E. coli still E. coli, and why haven't we morphed into one common bacterial genome, given the fact that microbes are constantly exchanging genetic information in a horizontal sense?
Because restriction enzyme modification.
Yeah, well, I understand. I was waiting. That's Elio's post today on yesterday on Small Things Considered was the restriction modification system.
Right. But I have a question. So typically in microbes, in general, particularly pathogens as well as environmentals, there's always gene duplication and backup systems, right? So if there are 80 of these things in Mycobacterium tuberculosis, it means that it doesn't really want to kill itself, right?
Yeah, well, that may be you may be right. That may be the point.
I mean, maybe the point that in case things get really, really bad and all 80 of them go out, you're done. But there's got to be some, like you're suggesting, some reason why. I mean, if it's duplicated 80 times, and if that's, I don't know whether that's genome sequencing or whether there are real products that come out of these, these modules. It just seems to me that it's going to be a little bit more, complicated than,
That's very true. Let me say that the number 80 comes from genomics. It simply comes from homologies. You have no idea if these things are really addiction modules or not. They may be or may not be. On the other hand, it's clear that they're not necessarily duplications. You're not duplicating the same thing many, many times over. All these are different. In the case of E. coli, the different addiction modules are in fact different.
Right, but I know that they're all different. They're not duplications, but they're backup systems that, you may be right. Michael may be right that they control different environmental cues, right? So I believe
that's very possible.
But it's got to be a global, I think what Michael suggests then is about a sporulation cascade. It's got to be globally regulated in some way if there are that many of them. But have people looked at protein expression in the test tubes? Even in mycobacteria, you could do this with,
sure.
PCR.
No, I'm not aware of much work. This is all fairly recent. I'm sure it's going to be looked at. Most of the work has been done in E. coli and a few other bugs. So we can expect that as I say, half the bugs seem to have them. So everybody's going to have to look for their favorite bug and consider this. It's just part of the new picture.
The other thing we should point out is some of these addiction molecules and toxins are effectively enzymes that actually cleave message inside the cell, which is, if you really think about it for a while, that's really a pretty remarkable concept in a organism that doesn't have a nucleus. That you invest the energy to make the message, and then you have an enzyme that can actually coordinate whether or not that message is actually going to be expressed. And I think in the case of, if you go back to tuberculosis and, it's got this really slow growth rate, TB makes its living by, just making a few messages every now and then. And I think that may also point to what is actually going on with tuberculosis. And since the enzymology of these molecules are actually RNA cutters, I think it's some are. And you can take a look at Masayoshi Inoue's work out of his laboratory, and he's done some really elegant work looking at the RNA activity, the RNase activity of some of these molecules. So it's really quite fascinating of mechanistically how these things do their thing and where their recognition elements and what they're actually targeting is just absolutely quite fascinating.
So, Michael, your work with going back, to the phage discussion we had last week, and you did some work. Was the goal of your work, not to plug your work, but it's really interesting now that I've heard about these things in a little bit more detail. So was the idea to deliver this particular cassette or module to a pathogen and then deregulate mRNA production or destroy or degrade turnover? Was that what you were doing, or?
We took a page, and Elio has already mentioned the name, is we took a page from Michael Yarmolinsky.
Right.
We started in phage, and some of this work was done with phage, and we were using it as a lethal agent to deliver to the bacterium without the antidote.
Wow,
so we would produce the phage, it would then inject the gene for the toxin, and the microbe would not have the antitoxin gene in it, and so it would go in and quickly kill the microbe. And you could use this on a infection model, where you would put in phage and it would effectively home out and seek the infection, and then take out the microbes without being able to have resistance emerge, as long as you had the receptor. But like everything, you depended on the phage to have the receptor. So we were looking for lethal agents, and these addiction molecules had been around for quite some time, and people had been using them for maintaining plasmids. As Elio points out, these addiction systems are truly wonderful for the fermentation industry.
So in this particular paper, they're looking at this, it's called MAS-EF. So MAS-F is the toxin which degrades the mRNA, and MAS-E is the antitoxin. So Elio, how does MAS-E inhibit the activity of MAS-F?
First of all, let me tell you about the name. MAS-E, MAS-F was discovered in Jerusalem at the Hebrew University in the lab of Ed Glaser and Hannah Engelberg Kulka. And they saw these genes being part of an operon that dealt with the stringent response. That's a subject we ought to take up some other time. Anyhow, they didn't know what they were doing there. And so they asked themselves the question in Hebrew, Ma-ze means, what is this? And the name stuck. So Ma-ze became Mas-E, anglicizing it a little bit. And the next one they called Mas-F because it was the next one over.
That's like the toll receptor story. You know, do you know
that's
that
right.
one?
Oh, yeah. That's a great story, isn't it?
Yep.
So anyhow, they saw these genes, and they as I say, they found that MAS-E binds to MAS-F, and when it binds to it, it inhibits it. That's the story.
Okay.
That's how it happens.
So then there's another player here, which is called EDF. Can you tell us about that?
Okay. Now this is a great story. It turns out that bacteria, in the E. coli at least, in this particular system, in addition to have this module, also makes a peptide called EDF, extracellular death factor, which works this way. It binds to MAS-F. So when it binds to MAS-F, the antitoxin can't work, and the toxin goes its merry way. So it's a lethal factor. Okay. It's the thing that I want. Imagine Margaret will want to pipe in on this. Is that of all things, it's a quorum factor. In other words, it's a molecule that tells the cell how many neighbors there are, how many cells there are around, and therefore it's expressed. The whole system is expressed when the population reaches a certain size, which is really fascinating. The peptide itself is kind of interesting. It's a symmetrical pentapeptide: asparagine, tryptophan, asparagine. And where did it come from? Well, it comes from the cleavage of a perfectly good, reasonable household enzyme, glucose 6-phosphate dehydrogenase. Now, you make something out of that. Nobody knows what that means. Why would this particular very decent, very conventional, respectable enzyme break up and make this peptide, which is then extruded? So this is a fascinating story, but it certainly points that the whole story is a population story. It has to do with the ability of cells to detect how many of them are around and kick in on that level.
Yeah, I think one of the things that, of course, comes to my mind immediately is that it's a kin selection story. And there has been some suggestion of kin selection in bacteria, but not very often. And kin selection is an evolutionary mechanism wherein there's a selection for behaviors that increase the inclusive fitness of the entire population. And so this is a fabulous story of kin selection. And I know that it's been, this is an altruistic sort of thing. And kin selection as an area of evolutionary biology was that phrase was coined by John Maynard Smith to describe what goes on in animals. And when they do something that would seem harmful to them, so that but it benefits the whole population. Has that been applied to bacteriology much?
I'm not aware of it. Is anybody else? I never heard of it, and it's not something I see in the list of possible functions for toxin antitoxin systems.
There has been, back in the old days when you're looking at bacteriocins. So the bacteriocin, story was, these proteins that were made that only the hosts that make them survive, and their other, other ones that don't produce them are killed. So it's kind of, a little bit different, but yet it will, do something good to select, people, not people, but bacteria that are like one another. Right. So it's kind of, that could be the forerunner of this, early discoveries, but that's the only thing that I know of. Could resemble something like this per se.
So I don't understand. So the idea here is that if the population gets too large, then this pentapeptide is produced, which then will kill off some of the members. Is that it?
That's right.
And so what senses that the population is too big?
Well, the concentration of this peptide. The peptide, the way quorum systems work is by making something constitutively, essentially, or, well, sort of. And then when there's enough of it, then something happens. Then the quorum sensor is activated and all hell breaks loose.
So the more members of the population that are producing it, the heavier the concentration. Is that how it works?
Yeah, well, the idea is, Margaret, why don't you give us your favorite examples?
Well, of course, quorum sensing was first discovered in my favorite bacteria, the luminous vibrios. Right. And that was, and so quorum sensing is a behavior that bacteria only do when they're in high numbers. And when they are at low density, they make this particular chemical at a very, sort of very low rate, and it just diffuses out into the environment. But when they get in very high concentration, that molecule begins to build up in their environment, and it goes back into the cells and interacts with the bacterial operon that's associated with light production. And there's a regulatory gene that is involved with this. But what happens is, at high density, the buildup of that molecule and the interaction of that molecule with the lux operon causes the light production of the bacterial population to turn on. So they turn on light production when autoinducer is at high concentrations and when the population is at high concentrations. And so it's thought that, one theory as to how it arose in the luminous bacteria is that perhaps luminescence evolved simultaneously with visual systems in animals, and that you could only, why would you want to be luminous if you're so small you can't be seen? But at high density, you're in a population that's large enough for an animal to see. And, in the case of when luminous bacteria have symbiotic associations, they're always in high population density. And so other animals can see the luminescence of bacteria that are in symbiosis with animals.
So in this case, this pentapeptide may be a similar situation. It's produced always at a low level, and then when the population grows to a certain point, it then somehow activates the endonuclease or inhibits the antitoxin.
Right. The point being that you don't want to waste your resources when there are few bacteria around. It doesn't make any sense to go through these motions.
Sure.
It doesn't make sense to make light. It doesn't make sense to go through this circuitry of altruistic suicide. It's also true when bacteria make toxins, they're often under quorum sensing control, because what's the good for 10 bacteria or 1 million bacteria or let's say 100 bacteria to make a toxin inside a great big host? But if you have 1 million or 100 million, then it makes sense.
Sure.
So it's a hedge your bets until it makes sense to bet.
Could I ask a naive question?
Sure.
So got Margaret. So if it's only, is it produced when the population reaches a certain density, and is that density then helpful to the population or deleterious to the population?
So in the case of, I can't think of a situation in which quorum sensing is deleterious to the population, except what we're talking about, this death factor. But in all the other circumstances that I know of with quorum sensing, in the case of the luminous bacteria, it's making luminescence itself is metabolically demanding. In other words, there's an estimation that about 5% of the metabolic potential of the cell goes into making luminescence. So there is a cost, but what they're doing is they're providing light for the host, and the host in turn feeds the bacteria.
Right.
So and in our system, if you make a deletion in the Lux operon, which is the autoinducer activated operon, if you make a deletion in that such that the bacteria are dark, the animal will not hang on to them. They know they're not doing their job, and they get rid of them. So there's a disadvantage to not doing it in that case. With things like Pseudomonas aeruginosa that make elastase or something like that in response to autoinduction in the lung or of a cystic fibrosis patient, then it seems that would be beneficial to them as well.
One of the interesting things, Cliff, is that this whole system of quorum sensing is actually manifested in strep pneumo. And the first genetics that most folks are introduced to in the microbial world is smooth and rough pneumococci. And natural competence is actually a quorum-sensed mediated event, in which rather than using a homoserine lactone in the gram-negatives, they actually use small peptides. Right. And these small peptides actually transform, no play on words, the strep pneumo so that it becomes competent to take up the DNA from the autolysine friends and neighbors in the general vicinity to ascertain whether or not there's any good genetic information out there that may actually facilitate it to live long until the next generation. So this whole notion of quorum sensing is really a pretty interesting story when you consider it globally of what it's doing.
Let me say a couple of reasons why the story we're talking about, which is really about death, about killing, it may be in fact non-detrimental, may be beneficial. But the first reason for saying that is just an ad hoc argument, a priori argument, that if a genetic system is retained over a vast number of bacteria and is present in multiple copies, there's got to be a survival reason for it. There's got to be a selective advantage to having it. So even not knowing what it is, you can say, if it's there in such numbers, it's got to be there for a good reason. But the second reason is that it does make some sense that these are systems for retaining certain large operons or integrons. It's also a system that may help in starvation, whereby cannibalism may in fact be a survival strategy.
Is it possible to use this antitoxin as a therapeutic target? So could you design molecules that say...
Michael was talking about this.
Yeah, right. Michael was trying to use that as a strategy to...
Right, so he was delivering the toxin with a phage, but could you design a small molecule that would, say, activate the toxin, make it constitutively active so that you would kill the bacteria?
The folks in Jerusalem have patented the EDF peptide for just that reason.
Good.
Right.
Good.
I don't know how far they've gone with it, but it's certainly a candidate molecule, or it points to a class of possible molecules. Peptides are really very popular these days, and so why not?
Sure.
And going back to Margaret's biofilm-based light organ, if you think about it, if you could design something that would chelate, if you will, the quorum sensing molecule, the homoserine lactone, when it's extracellular, such that it will not come back into the cell and turn on the genes to effectively enable the biofilm to form the light organ. That could effectively be a therapeutic in lung infection where Pseudomonas is involved, because Pseudomonas has a very similar quorum sense controlled operon that folks have been trying to design, if you will, a bait molecule that would scarf up all the inducers that the microbe is tossing out into the environment, so that even though there may be high densities of Pseudomonas present, they remain in a planktonic state rather than sitting and establishing the very difficult and challenging biofilm to remove from the CF child's lung.
That's a fascinating idea. We'll have to see if we can get something going in the squid. Yeah.
You have the ideal reporter molecule because
we do.
Can add the bait molecule external in the light organ and ask the question, is light made?
Oh, yeah,
and
absolutely.
Whether or not the biofilm actually, to use a immunological term, degranulate, if you will.
Before anybody gets really excited about quorum sensing as a potential antibiotic or therapeutic, there have been a number of companies that have come and gone that have been focused on using quorum sensing or inhibition of the homoserine lactone molecules, and it's very, very hard to get it to work.
It's all about delivery.
Well, probably, but the question is, where is the infection? And I would argue that if you want to, again, going back to what we were talking about, phages, I think where the biofilm situation may be the reason for the infection, like in a wound or something like that, diabetic foot, I think that would be the best way to test whether these things can work for those indications, because I think systemically it becomes very, very dicey based on the accessibility, how many bugs are really causing the infection, how diffuse the infection is. But I know that there was one company called Quorex that started back in the early 2000s, and that company is long gone, using quorum sensing, I think in Pseudomonas actually, as their target system.
Elio, anything else before we move on?
Well, you were going to ask me what my connection is with the PI of the Maziev.
Yes, I heard you had a connection with the PI. What is that?
Well, she's my cousin. And what's more, she's the brains in the family.
Oh.
Absolutely, no question about it.
Great.
So are you the patent agent that's representing the company in the U.S.?
No, I don't have a big family. I have two cousins, and half of them are microbiologists. So
wonderful.
Goes to show you.
All right, so that is Coping with Hard Times, Death as an Option. We'll put a link also to your post on Small Things Considered on that one.
Thank you.
The next paper was also suggested by Elio, and this is also very interesting. This is a paper in PNAS about cyanobacteria that fix nitrogen in the ocean. And this is really a cool story. Again, also, which I knew little about. This is a specific cyanobacterium, Crocosphera watsoni. Crocosphera, what a nice name. These are, I guess you know all about this, Margaret, right?
Oh, a little bit.
I'll give my summary, and then everyone can correct me. So these are marine cyanobacteria, and they fix nitrogen. They take atmospheric nitrogen, and they make ammonia out of it, which is important for the nitrogen cycle. And the interesting thing is that these organisms also carry out photosynthesis. They make oxygen when there is light present. And these are very contradictory processes in a way, because fixing nitrogen is not amenable to having oxygen around. So I guess the nitrogenase that's involved doesn't like oxygen. So how to regulate this paradox is an interesting problem. And so what they have done in this paper is, there has been some suggestion previously, I think, that there is regulation of of RNAs encoding the various proteins for nitrogen fixation versus photosynthesis. But what they did here is to look at the proteome of these cyanobacteria as they are growing through light and dark cycles. And proteomic technology is now at the point where you can take whole bacteria and break them open and look at thousands of proteins and ask, are they present or not? So basically you can take bacteria, break them open, then you treat the extracts with a protease to break up the proteins into small fragments, and then you put them in a mass spectrometer, and you can figure out the identity of thousands of proteins. So it's a good way to look at changes in populations, and it's being applied to all sorts of systems. And it's highly dependent on computational information, of course, because you basically need to have a sequence against which to compare your mass spec data. So what they have found here, interestingly, is that as the sun rises and the bacteria begin to do photosynthesis, they make the enzymes that they need to do photosynthesis. These are iron-containing enzymes. They do that through the day. And then as nightfall comes, they degrade those proteins and make new proteins that are needed to fix nitrogen. And they take the iron released from the photosynthetic proteins and use those for the nitrogen fixation. So there's this turnover every 24 hours where they make new proteins. They use them for one process, either photosynthesis or nitrogen fixation, and then they degrade them. So they say in the paper, this is a huge energetic cost, but it's probably something that has evolved because iron is limiting in the oceans.
Mm-hmm.
It's because they made all that damn oxygen. Because what happens, the thing that you need to remember is that the iron that is useful is Fe+2.
Mm-hmm.
And what these wonderful cyanobacteria have done for us is they've made oxygen. They've made Mother Earth an oxygen-containing planet, which is good news for us, bad news for iron-containing enzymes, because what happens is anytime you have Fe+2 out there in the presence of oxygen and water, as we all well know, you make rust.
Mm-hmm.
And you get Fe+3, which is insoluble. And so the life of the poor bacterium is not easy. And so they've learned that this is an exercise in sustainability that I think, we as a sentient species really need to pay attention to, because this reuse of iron has actually increased the productivity of the population, these authors estimate, by at least 40%.
Yeah, that's right. So the limitation of iron is emphasized. My colleague Dixon Despommier said if there are ships moving through waters and they're dropping iron into the water as waste, there are huge blooms of these cyanobacteria that form wherever the iron is put. So if you have runoffs from land with iron contamination, you get huge growth in these bacteria as a consequence. So these are among the oldest bacteria around. The cover of Microbe magazine from February had this image of a slice through a rock showing layers of a fossilized stromatolite. And each layer of this fossil was a covering of blue-green algae, which is what cyanobacteria used to be called, I guess, that captured sediment and became mineralized. So these are 3.5 billion years old, the oldest fossilized remains of life on Earth. So you're right; they were around a long time. They made oxygen for us, Michael.
Yeah, and I think that's what we don't appreciate. And the other thing you have to appreciate is even humans perceive oxygen as toxic, because how we transport oxygen in our system.
Yeah.
We put it into an iron-containing box called hemoglobin.
That's right.
And as it moves through our body, we only open that box very quickly, let the oxygen out. It immediately combines with those electrons to make water, because that's what all these antioxidants are all about, because oxygen is so toxic to membranes and proteins and DNA that we really try to contain it. And I think what this story is teaching us is we're beginning to develop tools to really begin to measure proteins in a temporal manner that allow us to detect these really remarkable relationships that the microbes have in terms of how they control their gene expression.
Yeah, the technology here is amazing. You couldn't have done this 5 years ago. And, you can look at RNA levels, but it doesn't really tell you what's happening with the proteins. And that was the key here, that these proteins are made, then they're broken down. And every 24 hours, it's amazing.
Well, I think that's the most important statement in the paper for a first-year graduate student. Transcripts do not measure protein abundance.
Yep. Yeah.
Margaret, I was wondering if what you think about the usefulness of this, cyanobacteria being used or being considered on a very large scale for making fuels. And in the blog, by the way, the blog on this paper was written by two students from the Golden's laboratory at UCSD, Susan and Jim Golden. And they said, I think very nicely, since large-scale industrial production is done mainly in outdoor pools, it will be critical to understand how unique day and night metabolic processes affect end products. What do you think?
Yeah, so one of the things that struck me here was actually two things. One was this propensity for all forms of life to have diel rhythms, circadian rhythms. And, the Goldens have done a phenomenal job at talking, about bringing these to the fore in microbiology. And circadian rhythms had been described very, very well in lots of animals and plants. And then people like Woody Hastings began to study them in cyanobacteria and in dinoflagellates and things like that. And now we're getting down to the molecular mechanism. So this being able to look at how the proteome changes on the day-night cycle, I mean, just phenomenal piece of work. I was really very taken. I think the other thing that was it Michael that said about the oxygenic atmosphere and so on, and it's a phenomenal story because oxygen, the bacterial enabling of the oxygenic atmosphere at just about every single evolutionary milestone is tied to that. And so when you think about the evolution of the eukaryotic cell, when a mitochondrion is, so to speak, invented, or there's an endosymbiotic event, captured.
Yeah.
That's the good way to say it. There's an endosymbiotic event. That's all about oxidative phosphorylation, and the beginnings of using oxygen as a terminal electron acceptor, but the oxygenic atmosphere not only enabled lots of the evolutionary events, including the water to land transition, and so the activity of these microorganisms in the ocean created an atmosphere that up in the upper reaches of the atmosphere, an ozone layer was created as a result of all of the oxygen up there and the interaction of the oxygen with sunlight and whatnot. And that was absolutely critical for the water to land transition so that animals and plants could grow on land. And so, these cyanobacteria and their behavior as the major players in this have been absolutely critical for the entire history of the biosphere. So this is fun to see them get the proteome and mechanisms at that level.
Why do cyanobacteria make oxygen?
Well, that's sort of an interesting answer. It's because they ran out of sulfur. They ran out of reduced sulfur, and they had to find another electron donor. And the electron donor they found was effectively water. They went from H2S to H2O. And the net consequence is the waste product for the cyanobacterium is actually oxygen.
Okay. So they need to make energy from the photosynthesis process, right? They needed an acceptor and a donor and it became oxygen.
A donor, and it became... So they have the two-part photosystem as opposed to the Z-scheme photosystem, which is the hallmark of bacterial photosynthesis that Howard Guest worked out a long time ago with his Z-scheme of photosynthesis.
So oxygen-using organisms are an accident, really, right?
No, they were created out of necessity because the planet ran out of reduced sulfur compounds.
Yeah, I mean, because it ran out of sulfur, and so they were made. But if we hadn't run out of reduced sulfur, we wouldn't be here, right?
That for us, that's true.
If the volcanoes had kept going, I suppose, well, we wouldn't want to be here either, right?
No.
So it's amazing paper. I love this. I asked someone here, and maybe you know the answer to this, Michael. Do cyanobacteria ever cause human infections?
Unfortunately, the answer is yes, and it really depends on how well your immune system functions. This is clearly a zebra type infection that most physicians wouldn't anticipate. But as with everything, if you're immune compromised, they can get into the wrong spot and cause problems. They are gram-negative after all.
Yep. Okay, that should do it for those two stories. Let's do a couple of emails before we wrap this up. Let's see here. We have another one having to do with nanoparticles. This is from John. He asks, is the effect of silver nanoparticles related to electron transport? What determines which metals will be antimicrobial, copper, silver, and which won't be, iron? I think Michael could do that.
Well, the simple answer to this question is it's all about their electron shells and how electrons move. And the antimicrobial activity of silver is attributed to the silver ion, the Ag+, while the copper mechanism is a little bit different. And the mechanism for copper hasn't yet formally been worked out. There have been a number of papers that have been published on that subject. Silver, we know from some elegant clinical trials, it's really a silver ion that is responsible for the inactivation, and what it principally does is it interacts with the sulfhydryl groups of enzymes and renders them inactive because it effectively complexes with the sulfhydryl.
Well, Michael, you know something about everything.
He does. I was just thinking the same thing.
I was trained as a microbiologist, and in order to survive qualifying exams, I had to do that.
Gee, I didn't have to do that.
I had the same experience, so I kind of...
And we went to those fine Big Ten universities where they made you real microbiologists.
Biology, not fake ones.
Where did you go again, Michael?
The amazing thing is you remember all this.
I got my graduate training at Indiana University in Bloomington.
Okay.
But the amazing thing to me is you remember it all.
Yeah. It's an interesting experience.
Elio, he'll start to forget soon.
Okay.
Maybe in the lifespan of TWiM. We'll see. Okay, the next one is from Krista, who writes, I'm a medical laboratory technologist working in clinical microbiology at Mount Sinai Hospital in Toronto, Ontario. I'm an avid listener of TWiV and TWIP and have been hoping you would start a bacteriology-themed podcast for a very long time. The first few episodes of TWiM have been fantastic. As a recent graduate and novice technologist, your podcasts are an excellent way for me to build knowledge and to keep current on the infectious disease literature. Now I just have to find a way to get my accrediting body to give me continuing education credits for listening. Have you ever considered getting your podcast CME accredited for the physicians out there? I don't know how to do that. Do you, Michael?
Oh, it's a chore.
Is it really?
Yes, I know how to do.
You need learning objectives, and you need to give questions per however many minutes, and it's a chore. But there are a lot of good online CME opportunities out there as well.
Yes.
Okay, keep up the great work. A big thanks from Canada for TWiM. The next one is from Suzanne. TWiM is an amazing podcast. I'm a pediatrician from the root of PhD in molecular genetics, transformed into an MD, and now working in the world of primary care pediatrics. Microbiology is 99% of my life. Your podcasts are joyful. Basic observations, research, molecular mechanisms, insights, perspectives, bridging the basic science to the possibilities in medicine and patient care and healthcare policy. You have filled a niche. Gut flora hit home. Probiotics were added to infant formulae in Europe long before here in the U.S. Yogurt and probiotics have been shown to lessen antibiotic-associated diarrhea. The ideal age to introduce solids is 6 to 7 months. Why? Gut flora, simple and beautiful. I chose to become a pediatrician after PhD training and a 5-year postdoc to bring basic science to children to hopefully improve their lives. Your podcast satisfies my basic science core and provides me with evidence-based insights into healthcare. Thanks. My ideas for future topics are 1, what is the epidemiology that has so dramatically changed the virulence of strep and what's up with a vaccine? 2, upper tract infections help. What's one to do with multi-drug resistance? And 3, if all disease has a microbial relationship, fill me in on autism and mental illness. From the trenches.
Well, urinary tract infections are, the issue there with multi-drug resistance. It's, again, simple selection pressure of making certain that when patients are given antibiotics, they take them for the full course so that you don't get the emergence of the resistant strain and you take them at the appropriate strain. And again, antibiotic susceptibility testing is really very important for UTIs to make sure you just don't do it with empiric coverage, but the antibiogram of your community. And so get to know your public health people because they'll tell you what's running around your neighborhood. Most of your referring hospitals or hospitals in your area have antibiograms, so you'll know what the garden variety UTIs like E. coli, and E. coli is the principal UTI out there. They'll tell you which drugs work in your particular area. So if you just do empiric treatment of UTIs, you'll hopefully get the right one so you won't get the emergence of resistance. So okay. It's a big problem with multi-drug resistance, and we're seeing that across the country. And you just turn on the news some nights, and you see about these multi-drug resistant microbes evolving out of hospitals and now in the community as well.
Okay, our next one is from Dr. Tom, who writes, long-time Twipper, soon to be a long-time Twimmer. Love the information. Thought that a podcast on probiotics would be timely. Seems like I, as a pediatrician, am suggesting them more for a multitude of ailments from recurrent abdominal pain to acute gastroenteritis to antibiotic side effect amelioration. Also, it is getting harder to purchase infant formulas without some form of prebiotic or probiotic. Keep up the great work from snowy and cold Madison, Wisconsin. I guess we got this a month ago. Probiotics, 2 in a row, huh? Okay.
Well, they're having a session at the General Meeting of ASM on some aspects of probiotics, so we can go and visit the sessions and see what the hot stuff is.
We will. Good idea. What else to do in New Orleans, right? All right, we'll read one more. This is a good one since Margaret is here. I'm interested to know, this is from Peter, I'm interested to know if it is generally true that pathogenic microbes produce biofilms. Also, in a polymicrobial infection, what are the ecological interactions between the bacteria? Do the biofilms produced by one species protect or inhibit other species? Could a normally non-pathogenic bacteria become harmful if combined in a biofilm with another organism? I'm asking this out of interest. I am a photographer and not involved in any medical research. I do have a long-standing interest in science and find your podcast to be fascinating.
Wow.
Is it true that pathogenic microbes produce biofilms?
Yes.
Yeah, we've touched on that earlier in today's story by talking about the Pseudomonas aeruginosa that forms the biofilm in the lung of a cystic fibrosis patient.
Right.
Oftentimes mixed infections or polymicrobial infections do form cooperative biofilms, and so that's everyone's nightmare. You can actually do this experiment at home. Don't brush your teeth for a few days, and you can actually experience the polymicrobial biofilm that develops in your oral cavity. And you can actually experience the plaque formation that is a consequence of the biofilm being laid down, and it's a a polymicrobial gram-negative, gram-positive based infection that is responsible for the plaque that develops on your teeth.
So do biofilms made by one species protect or inhibit those of another?
Well, there's competition, and those same quorum sensing molecules that are out there, and I'll bet if we begin to do some of the experiments with these death factors, we'll see that could probably lead to the dynamic equilibrium that is omnipresent in these polymicrobial biofilms.
And the last one, can a non-pathogenic bacteria become harmful if combined in a biofilm with another organism?
The answer is yes as well, because they are responding to the environment. And the classic way of doing that experiment is you take a pathogen out of the patient and you pass it in successive cultures on a Petri plate in your lab, and you sort of tame it. Similarly, the way you make it virulent is you put it back into the animal, and passage through an animal often allows it to regrow its fangs, if you will, and become a nasty pathogen once again.
Okay. There are many ways to listen to TWiM. You can find us on iTunes, at the Zune Marketplace, or at microbeworld.org/TWiM. We also have an app for your iPhone or Android device at microbeworld.org/app. If you like TWiM, go over to iTunes and leave some comments. It helps keep us high on the lists of medicine-related podcasts. Send us your questions and comments to twim@twiv.tv. I'd love to thank everyone for being here today on this Friday the 13th. Margaret, University of Wisconsin, thank you so much.
You're most welcome.
And I think you're going to be in New Orleans next week as well, right?
I certainly am.
All right. Well, hopefully we'll run into you.
Yes.
As Michael is as well. Michael from the Medical University of South Carolina. Thank you.
Thank you, Vincent.
Elio, Small Things Considered, thanks for joining us again.
My pleasure. I won't be in New Orleans, unfortunately.
I know.
Elio, you're not coming.
No.
I'm disappointed.
Well, I feel the same way. Sorry. Can't make it this year.
Okay. Well, we'll have you next time. And Cliff Mintz is at biojoblog.com. Thank you, Cliff.
You're welcome. And who's ever going to be in New Orleans, go to Couchon.
What's that?
It's a restaurant. It's pig in French.
All right.
So go there and tell Matt and Tyler that you know me, and they'll take care of you.
Oh, either they'll take care of us or throw us out.
Well, they usually are very nice to me, but I can't vouch for what they'll do.
You bet. And don't forget TWiM Live at the annual meeting of the American Society for Microbiology in New Orleans, Louisiana. That'll take place at 2 p.m. on Sunday, May 22nd. Room 232 of the Ernest Morial Convention Center in New Orleans. And we have 6 guests in addition to myself, including Stan Malloy, Michael Schmidt, Nicole Dubillier, Andreas Baumler, David Aronoff, and Paul Rainey. And it will be live streamed on Ustream and, of course, recorded. So you can watch it live, or you can wait till it comes into iTunes. And if you want more information, just go to microbeworld.org, and you'll see on the front page on the upper left a badge for ASM Live, and you can get details about the sessions. So we'll see you in New Orleans next week. I'm Vincent Racaniello, and you can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, Communications Director Barbara Hyde, and Chris Kondayan and Ray Ortega for their technical help. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.